Good morning, everyone. Welcome to the webinar for March. My name is Rebecca Bailey, and I will be the moderator for today's webinar. Today's speaker is Dr. Amy Moody. Dr. Moody will be answering questions at the end of the webinar. You can submit your questions at any time during the webinar by entering them into the questions box on your GoToWebinar side panel. So if you have anything to ask, go ahead and enter it there. Dr. Amy Moody is a senior principal scientist in the Translational Modeling and Simulation Group at Pfizer. She earned her Bachelor of Arts at Kenyon College and her PhD in Cellular, Molecular, and Developmental Biology from Tufts University. Amy began her scientific career as an experimentalist, and in her PhD, she studied the role of channels and membrane potential in stem cell differentiation and cancer. As part of this work, she used silk as a scaffold material to develop improved 3D tissue models of breast cancer that could be cultured in vitro for multiple months. She then transitioned to modeling during her postdoc at Tufts University and at Dana-Farber Cancer Institute with support from the Lab of Systems Pharmacology at Harvard Medical School. Her postdoc project involved investigating macrophage immune signaling dynamics using a combination of experimental and modeling approaches. Amy joined the Translational Modeling and Simulations Group at Pfizer and has primarily supported small molecule rare disease programs. She's developed highly mechanistic mathematical models of pharmacology that have enabled key insights and support decisions at multiple phases of R&D. It is with great pleasure that I welcome today Dr. Amy Moody. Amy?
Thank you so much. Thank you for that kind introduction, and thank you to Rosa for inviting me to give a talk today. I'm very excited to talk through a couple examples of some modeling work in the preclinical space supporting the development and progression of preclinical programs. Let's see. So first, the requisite conflict of interest statement. All of the work that I'll be presenting today was funded by Pfizer and was performed entirely by Pfizer employees. And so today I'm going to be talking about two different programs. First, Tafamidis follow-on program, and then some modeling work that was done to support sickle cell disease programs. And the modeling approaches were really quite different. In the first case, using clinical data from a large cohort, individual data to drive the model. And then in the second case, a much more theoretical model. But in both cases, I hope I'll be able to show today that these very different approaches were able to give a really good sense of the likelihood of success for programs that helped with decision making in terms of whether programs should continue, as well as prioritizing different programs. And I will say that I'm not going to go into a whole lot of technical detail today, but on my last slide, I'll provide references to publications where we've published on both of these approaches and where you can find all of the technical details. And I would be happy to follow up with anyone by email if you have questions along those lines. So first, to introduce you to Tafamidis. Tafamidis was the first FDA-approved drug for amyloid transthyretin cardiomyopathy. So this is a disease that's characterized by amyloid formation that is based off of pieces of TTR or transthyretin protein. And so in the schematic here is showing what's generally accepted as the way that this disease occurs, where TTR is produced in the liver as a tetramer and is secreted into the plasma as a tetramer. That tetramer can dissociate first into dimers and then into monomers, and those monomers can become misfolded. And the current thinking is that it's primarily the misfolded monomers that are driving amyloid formation. And so by stabilizing TTR and keeping it in the tetrameric form, that's thought the hypothesis was that that would decrease progression to the monomer and therefore misfolded monomer and decrease the rate of disease progression. So as I mentioned, Tafamidis was the first FDA-approved drug in the class of what are known as TTR stabilizers. And so these drugs bind to one or both of the thyroxine binding sites on TTR and stabilize the tetramer in its tetrameric form, essentially inhibiting dissociation. And so that really drives this pathway. It shifts the equilibrium so that more of the protein is in the tetrameric form, which has the impact of both reducing the amount of monomer and therefore misfolded monomer, as well as because by preventing the formation of monomer and making it so that less of the protein goes through this degradation pathway, it also has the effect of stabilizing, or sorry, leading to accumulation of the tetrameric form in plasma. And so the increase in TTR associated with that is actually one of the first measures of pharmacology. And so the story of Tafamidis provides really nice confidence and rationale for this class of molecules because Tafamidis was able to significantly reduce mortality as well as to reduce a number of markers of disease progression. However, while Tafamidis was quite efficacious, it was not fully efficacious, that the disease does continue to progress. And so that leads to an important question. How well does Tafamidis stabilize TTR in the clinic, and can it be improved? And at the time that I was tasked with helping to answer this question, there were some conflicting reports in the literature of the level of TTR stabilization that Tafamidis achieved, as well as a competitor that was entering the clinic with claims of achieving greater stabilization. And so here below, I'm showing a few of the different data sources that were available at the time. The first two represent biochemical assays that were performed. Here, acidic denaturation assay with a comparison of Tafamidis in blue versus the competitor AG10 in red. And in the second, a similar study that measured TTR stabilization, in this case under physiological conditions, again with the competitor AG10 in blue here and Tafamidis in red. And so you can see is that, first of all, in both of these cases, they're reporting that the competitor provided better stabilization than Tafamidis, but the assays give fairly different values, with the version of the assay presented here showing a much bigger difference than what's presented over here. And so there are important questions about whether this assay that's performed under acidic conditions, is that really relevant to the physiological condition? And then the other important aspect is that none of these studies use the clinical concentration of these two compounds. And so here, the concentration of Tafamidis was substantially below the level that's reached in the clinic. And again, questions remain about, what is the clinical level of stabilization? Can these assays tell us anything about that? And then finally, the last set of data over here on the right is looking at the change in TTR levels associated with each of the drugs. In the case of Tafamidis, reporting it out from the phase 3 trial. In the case of the competitor AG10, it was based on a smaller study, an early phase 1, phase 2 study. And so looking at this data, recall that as I mentioned in the prior slide, the increase in circulating TTR is a direct indication of TTR stabilization. And on the one hand, looking at the wild type patients, it's somewhat similar between the two drugs. However, looking at the variants, it does appear that AG10 provides greater stabilization. However, it has to be considered that the level of TTR increase is a function of both the TTR stabilization, but also the basal level of dissociation of the tetramer. And so what I didn't mention before is that amyloid patients comprise two groups, one that's made up of patients that carry wild type TTR and one that carry genetic variants where the variant leads to a faster dissociation rate of the tetramer. And with the faster dissociation rate, stabilization provides a greater increase in circulating TTR. And there are a number of different variants, quite a few, each of which will have a unique dissociation rate. And again, the greater the dissociation rate, the greater the increase in TTR. So while this on first view does appear to be a significant difference between these two compounds, what we can't tell from this data is whether this is being driven by greater efficacy of the drug itself or whether it's a difference in the patient population. So we wanted to analyze the data to the greatest of our ability to provide a better quantitative understanding of TTR stabilization that is achieved by Tafamidis. And so to do this, we tried to break the question down into 3 easily answerable questions. The first, what is the occupancy given the known clinical exposure of Tafamidis? How much of site 1 is bound? How much of site 2 is bound? Secondly, how much stabilization does the first and second occupancy of the first and second site provide? We knew from the literature that based on ex vivo studies, people had reported that binding of the first site was sufficient for complete stabilization, but could we find evidence in the clinical data to support that that was true in vivo as well? And then finally, based on our answers to the first 2 questions, what did that say about what was left in terms of additional horsepower that could be reached by a similar mechanism? How much could Tafamidis be improved? And so to answer this question, we first needed to develop a binding model to give a calculation of occupancy based on the concentrations of TTR and drug in the clinic. And so since Tafamidis was approved at that time, it was of course very well characterized. And so what we knew is that Tafamidis bound the 2 sites of TTR with negative cooperativity, meaning that for the unbound TTR, there were 2 equivalent sites with the same dissociation constant, KD1, but upon binding of one of the sites with Tafamidis, it led to a conformational change of the second binding site that changed the dissociation constant such that it reflected KD2, a higher value. We also knew that Tafamidis had relatively high affinity for albumin, and therefore that in order to come up with an accurate assessment of the equilibrium binding condition, we needed to also account for this binding to albumin. So although we knew the structure of the model, we didn't have good estimates of the affinity values at this time, and so we needed to generate data to help provide those values. And one of the challenges that we ran into is that it can actually be somewhat difficult to find assays that have sufficient sensitivity to KD1 and KD2 when dealing with negative cooperativity that's small to moderate in size, but not extremely large. And so I'm just trying to give an example of that here, looking at ITC data on the left and some SPR data on the right, where the data is the same in each plot, the data represented by the dots, but the model is fit either with a single KD value for site 1 and site 2, or with a negative cooperative model where KD1 and KD2 differ. And what is, visually, it's not apparent that the 2 models are much different, that if you were to just glance at this quickly, you wouldn't immediately jump to the conclusion that the negative cooperativity model is actually the right model for the data. However, with appropriate statistical analysis and looking closely at the objective function values, when you fit the data, you can demonstrate that, in fact, that is the correct binding model. But again, it takes some effort to really find assays that are sufficiently sensitive. And so we think that that partly explains why if you search the literature for both Tafamidis and other TTR binders, you will actually find quite a number of publications that reference a single KD, but we don't believe that's accurate. And so we also relied on an assay known as the subunit fraction exchange assay. And this assay was very helpful because it was especially sensitive to KD1. And so this assay, as the name implies, is looking at the degree to which the different subunits of TTR are exchanged. And so in this assay, you begin with 2 pools of tetramer, one pool in which none of the subunits are tagged with a flag tag, and a second pool in which all 4 subunits are tagged. And in this assay, using HPLC, the difference in the weight driven by the tag allows you to separate all the possible species of tetramers. And so as you incubate the sample over time, the tetramers dissociate and then reassociate. And when they reassociate, with equal affinity between the tagged and untagged version, the species, all of the species, possible combinations of tagged and untagged will form over time in the ratio that you expect based on probability. And so in running this assay, multiple time points are measured to assess the fraction exchange over time. And this time course data can then be fit to a rate constant that is the apparent dissociation constant of the tetramer. And the other point that I want to make about this is that, as I said, what's really nice is that the output of this assay is a rate. whereas much of the data in the literature is based off of assays that work in a similar fashion, where what you have is a nonlinear change in time of the amount of tetramer that remains in the same tetrameric state. And so you can see from this that whether you collect your data at day 2 versus at a later time point, you would get a very different absolute amount. But the rate, if you take account for the whole time course, you get the rate, which is actually the most important value. And so, this assay has been described in the literature by a couple of different groups. And what they found looking at different concentrations of different compounds is that the apparent dissociation rate is equal to the base dissociation rate of the tetramer times the fraction unbound. And that's shown below here in the relationship between unbound tetramer versus the rate. And this is one of the assays that provides data that binding of a single site is sufficient to give complete stabilization, at least in this ex vivo assay. So getting back to our binding model, what we found was that it was necessary to combine data from multiple experiments to be able to get an accurate prediction of the KDs. And so we used a combination of the subunit fraction exchange assay shown here that was very sensitive to KD1, as well as ITC data, and specifically ITC data performed at multiple concentrations of TTR. That was very important to make the data sensitive to KD2. And then finally, as I mentioned, we knew that Tafamidis had high binding to albumin, and that getting a good estimate of the dissociation constant for albumin was also important. And so we based that off of a concentration curve fraction unbound with albumin data. And from that, we got really nice estimates of the KD with 5 nanomolar KD1, 200 nanomolar KD2, and then a much higher 3.5 micromolar KD for albumin. And so we took that binding model and we applied it to the individual data that was measured in patients. And it was very nice that from the patients, we had the drug concentration, the total TTR concentration, and the albumin concentration. So we had, we didn't have to make assumptions about population distribution of the protein levels. We had actual individual measurements from the patients, which we felt was very helpful for getting the most accurate estimates possible. Also, in the patient data that we had 2 different dosing cohorts, 20 milligrams and 80 milligrams. And that was really nice because, as you see here, we get quite a wide distribution of occupancies across the whole population. And so again, applying that binding model to all of the patients, we were able to calculate the occupancies. And today I'll be focusing on the 80 milligram dose because that was what was chosen as the clinical dose. And from this, we determined that site 1 was on average occupied at about 94%. And I should say site 1 represents all of the TTR that has at least one site bound. And then site 2, that's the only representing TTR where both sites are bound. And at the 80 milligram dose, that's about 9%. And so this aligned nicely with what the team already knew. They knew based on that negative cooperativity and the concentrations that they were achieving that they were likely getting high binding of site 1 and low to moderate binding of site 2, but it was very nice that with this quantitative analysis, we now had a much better understanding of exactly what level of occupancy we had. And so then the next question to ask was, again, this question of in vivo, which of the sites provides stabilization? Is site 1 on its own sufficient, or can we also find an indication that site 2 is providing additional benefit? And to answer that question, we first started by looking at data from patients of the change in TTR with Tafamidis treatment. So again, as I mentioned earlier, with TTR stabilization, it leads to an accumulation of tetrameric TTR in plasma. And you can see the dose responsive effects shown here, where time 0's on the left and then multiple years into dosing. And you can see that increase on both a population and individual level. And so we built a model to describe circulating TTR levels. We kept the model pretty simple, but still captured what we believe to be the key biology. Having a synthesis rate for the tetramer, then the tetramer has a dissociation to the monomer, and then both the tetramer and the monomer can be degraded at different rates. And we accounted for the effect of Tafamidis by having inhibition of the dissociation. And as I'll describe, we tested a couple of different structures of the model. One in which dissociation was completely inhibited by binding of a single site, but also other models that allowed for intermediate level where perhaps single site binding reduced the dissociation rate, but only by a smaller degree, and then having perhaps only the second site completely inhibiting dissociation. But what we found when we analyzed the data was that the best fit of the data was the case where single site binding was sufficient to completely inhibit TTR tetramer dissociation. And again, we tested a couple of different models, and in each of those cases, adding additional parameters did not lead to an improvement in the model fit. One aspect of the model to consider, we did have to separate the wild type versus the variant patients. Again, the variant patients have a faster basal dissociation rate, and that allowed us to capture the effect where the variant patients actually have a greater % increase in TTR. And so here, the model predictions are shown by the green and blue lines, where green represents wild type and blue represents the variant. And so that's especially obvious in the case of the 80 milligram dose, where you see the variants have a much greater increase in TTR than the wild type patients. So again, captured the data really nicely, and in all of our different attempts to explore whether binding of the second site provided additional stabilization, we did not find that that improved the model. We performed a similar analysis on other measures of disease progression, including pro-BNP, the KCCQ questionnaire test, and the 6-minute walking test. And in all three of these cases, we ended up finding the same thing. We found that the effect of Tafamidis was best described as a linear relationship to site 1 occupancy, and that testing a model where site 2 provided additional occupancy did not lead to an improvement in the model fit. So in sum, we interpreted all of this analysis to suggest that the second site has negligible impact on TTR stabilization and on improving disease progression. So just in summary of what we found, we developed this binding model and applied it to the clinical data for Tafamidis, demonstrated that single site occupancy is sufficient for TTR in vivo as well as had been reported in vivo. Based on our modeling of the occupancy, we found that the Tafamidis 80 milligram dose provides on average 94% site 1 occupancy, which another way of saying that is that it stabilizes 94% of TTR. And for that reason, there's really minimal ability to improve upon Tafamidis by this specific mechanism of action, given that the greatest you could possibly achieve is 100% TTR stability, and are finding that there's a linear relationship between occupancy and efficacy. We took that to indicate that there's really very minimal room to improve upon this compound. And so far, that prediction seems to be playing out. AG10 did go on to complete a phase 3 study where it did not perform substantially better than Tafamidis. And I want to just talk through another aspect of that was sort of unique to this program that had important implications, and that was the impact of a high target concentration on PK. So TTR is found in relatively high abundance in plasma. And so what we found, and the model also predicted, is that there's concentration dependent fraction unbound in plasma. And so here the observed data represented by the gray dots, as well as the prediction provided by our binding model. And this is what one would expect given the scenario that at very low drug concentrations where occupancy of the target is relatively low, almost all of the drug sticks to the target, leaving very little of it to be free. But then as the target becomes saturated, more and more of the drug ends up being free in plasma. And so you get this shift from the low value to a higher value that's driven by the affinity to albumin. And so this has important implications for PK, because pretty much all of the equations that are sort of your standard PK compartment type equations are based on the assumption that fraction unbound in plasma is constant. But when fraction unbound in plasma is concentration dependent, then clearance, the blood to plasma ratio, the volume of distribution, and the half-life are all concentration dependent as well. And so in considering the PK of the molecule, we developed a model that accounted for all of this. And to do that, we used a PBPK approach. And so the tissues that we focus on are the typical set that one would use for a minimal PBPK type model. But then within the blood compartment, we explicitly included the binding reaction. So in plasma, we have the binding to the 2 sites of TTR, as well as the binding to albumin. And by doing that, we then track as concentration changes, we're tracking the actual free concentration, and thus are accounting for the differences in clearance, et cetera, because only the free drug is available to be cleared. And so one of the interesting aspects that came out of that analysis is that actually it's really important the level of albumin binding and the relative affinity for albumin versus TTR. And so I'm showing this here with a simulation of hypothetical compounds where the compounds are identical in terms of potency, clearance, other properties, but the one difference is the affinity to albumin, where the dark blue line represents a high affinity to albumin, whereas the light blue represents a low affinity. And what you can see here is that there's a dramatic difference in the peak to trough between these two compounds, where the low affinity compound has a much bigger peak to trough in terms of concentration. And of course, that translates to a bigger peak to trough in terms of occupancy as well, which, I think for many indications, it's preferred to keep as steady of occupancy over the dosing interval as possible. And so the explanation for that is the difference in the fraction unbound. And so as I said before, as the target becomes saturated, then more and more of the drug is free, and it's moving towards an upper limit on the fraction unbound that's driven by its albumin binding. And so for this lower affinity, this compound that has lower affinity for albumin, it's much more free. And so there ends up being a much bigger shift between the fraction unbound when it's being driven by Tafamidis or when it's being driven by the target binding versus when it's being driven by the albumin binding. And so what we found is, as I mentioned before, there's kind of this ideal level where the affinity for albumin needs to be substantially less than the affinity for TTR. Otherwise, the albumin will compete the drug off of the intended target, but also sufficiently high so that more of the drug remains bound, which protects it from clearance, and therefore extends the half-life, which is actually a very nice property of the drug. And one of the reasons that I bring this up is that some of the competitor molecules referencing Tafamidis have described the high binding of Tafamidis to albumin as a negative trait. But in fact, that is driving actually the really nice PK of Tafamidis, that it has a long half-life and a very small peak to trough. Okay, with that, I'm going to shift gears and talk about the other project. And so this was some modeling work that was done to support a few different mechanisms of action related to sickle cell disease treatments. So first, just a very quick introduction to sickle cell disease. It is a disease that is caused by a point mutation in hemoglobin, the protein that is responsible for carrying for carrying oxygen within red blood cells. And one of the interesting parts of this mutation is that it enables polymerization of specifically the deoxygenated form of hemoglobin, as is depicted here. When these polymers form, they're very stiff, which causes the red blood cells to deform and take on this sickle-like shape. And that leads to two negative outcomes for patients. The first is that the cells have a shorter half-life. They're recognized in the spleen as being improper and therefore turned over more quickly. And then second of all, they can become stuck in capillaries, preventing blood flow downstream of wherever the blockage occur. This is known as a vascular occlusive crisis. They're often extremely painful, and they can lead to severe acute effects such as stroke. But even the smaller events lead to a micro death of tissue, and over the patient's lifetime, unfortunately, this accumulates to the point of causing organ damage. And even today, for these patients, sadly, the life expectancy for these patients is only in the 40s and the 50s. And another interesting aspect of the polymerization process is that it can be described by two stages. First, nucleation, which is an energetically unfavorable process and then continued polymerization, which is highly energetically favorable. And so due to those properties, you get what's shown here, where the time to the beginning of polymerization is a stochastic, somewhat stochastic process, and there's great variability. But then as soon as polymerization starts, it generally occurs very quickly. And another aspect that was discovered quite a number of decades ago is that the lag time of nucleation is highly dependent on the concentration of deoxyhemoglobin, such that, as shown here, for a relatively small change in concentration, there's a relatively large change in the average lag time. And so it's been postulated for quite some time that if you could decrease hemoglobin concentration by just 10, 15%, that would lead to a meaningful increase in the lag time. Keep in mind the red blood cells are in circulation, so they're only in the deoxygenated environment for a certain percentage of time. And if you could reduce the probability of a cell sickling, you would, in theory, lead to improvement in patient outcomes. And so today I'll be talking about 3 different approaches that all sort of converge on the same idea of trying to decrease the concentration of deoxyhemoglobin. The first is to decrease the endogenous allosteric regulator 2,3-BPG. So 2,3-BPG binds specifically the deoxygenated form of hemoglobin and stabilizes it in that state. So by reducing it, one would push the equilibrium towards a more oxygenated state. The second is to essentially provide the reciprocal effect, which would be to create a drug that stabilizes the oxyhemoglobin state. And then finally, I think one of the most well-known approaches is the idea of replacing the sickled version, or the adult form of hemoglobin, which carries the sickled genetic mutation, with the fetal form of hemoglobin. So this is the form of hemoglobin that's expressed early in development, but then switches to the adult form following birth. And this form does not contain the mutation, thus would not lead to disease. And so for a long time, we know from patient data that patients that endogenously continue to express 30% HbF or more are largely asymptomatic. And so that has kind of represented the gold standard of what's required to essentially functionally cure this disease. And that's also been shown more recently in some of the genetic approaches that are being used, some of the gene therapies that are either switching expression or replacing HbS with HbF, where when they're able to reach 30%, they see a dramatic improvement in the number of VOCs in those patients. And so all 3 of these mechanisms are in the clinic or in clinical development in various forms. But to date, none of them have really achieved transformational efficacy for the majority of patients. As I mentioned, the gene therapies are reaching a very high level of efficacy, but sadly, the predictions at this time is that they will only be able to treat at most a few hundred patients a year out of the thousands of patients. There are roughly 100,000 in the US alone, and millions worldwide. And so one of the questions that we wanted to answer was, first of all, given these other 2 approaches, can we determine the level of therapeutic intervention that would be required to achieve equivalent efficacy as 30% HbF? And then secondly, given the challenges that have been faced by all 3 of these mechanisms, and as is often the case, it can be very hard by a single drug to hit the target as hard as is preferable. What can we understand about combinations to see that, again, all 3 of these mechanisms have things in the clinic that aren't necessarily hitting the fully efficacious level, but are having some level of efficacy? Can we make predictions about what level of intervention is required in combination? And so to answer this question, I worked with others to develop a model of oxygen saturation. And so the key assumption of this model is that we can simplify this disease to just thinking about deoxyhemoglobin in the peripheral tissue. That if one assumes that polymerization and sickling is going to occur in the peripheral tissue and is a function of the concentration of deoxyhemoglobin, then we can conjecture that any patient that has the same concentration of hemoglobin again, deoxyhemoglobin peripheral tissue, whether they got there by HbF, 2,3-BPG reduction, or some combination of any of these approaches, that they are going to have the same probability of polymerization sickling, and ultimately, therefore, the same response in terms of VOCs and other measures of disease severity. And so I sought to develop a model that would include all three of these mechanisms. And I based the model off of the well-known Monod-Wyman-Changeux model of oxygen saturation. And the reason that I chose this model is because it's mechanistic, and so that made it easy to continue to build out the mechanisms to include the three mechanisms that I'm interested in adding. And so just quickly, the basis of this model is that hemoglobin can exist in one of two states, the R state or the T state, each of which has a unique oxygen affinity, where the oxygen affinity of the R state is much greater than the T state. The other key part of this model is that the equilibrium between the R and T state is dependent on the number of oxygen ligands that are bound, such that when little or no oxygen is bound, the T state is highly preferred, but when many oxygens are bound, the R state is highly preferred. So this model does not directly include cooperative binding because the oxygen affinity is the same for the two states, regardless of the number of oxygens that are bound. However, because of the difference in the equilibrium transition between the two states that is oxygen dependent, that gives it apparent cooperativity. And so the way that this model is typically used is that if you're interested in developing a model of oxygen saturation under a specific set of conditions, you run experiments under that set of conditions and fit the three key parameters of the model, which are the two oxygen affinity values and this L naught term. And from that term, if you assume that oxygen binding reaches equilibrium very quickly, which experiments have shown is a valid assumption, then you can calculate out, based on assuming equilibrium, the oxygen saturation. However, because typically the way this is used is that the parameters are fit to a set of specific conditions, it's difficult to extrapolate this beyond the conditions that were tested, and it can be very laborious to test all of the conditions of interest. And in this case, we are interested in, of course, every possible combination of HbF, 2,3-B concentration, and potential concentration of a oxygenated hemoglobin stabilizer. So as I said, my approach was to then extend this model to mechanistically include the other three approaches. And so the first thing was to add species that represent HbF to the model. And so based on what's known in literature, HbF is known to have higher affinity for oxygen than adult hemoglobin, and this is simply driven by the fact that it has no binding pocket for 2,3-BPG. And so showing down here, I built that rule into the model, and I'll get into some of the details later of how that was built out. But that led to the expected shift in an oxygen saturation curve that's previously been reported. And the assumption is that all other parameters between HbF and adult hemoglobin are the same. The other important consideration was what to consider HbF. And I bring that up because there's a number of publications to make it quite clear that it turns out that the fetal hemoglobin and adult sickled hemoglobin can associate to make a mixed tetramer hemoglobin species. And this is really well described in the literature. However, somehow it ended up in sort of a pocket of the biophysical hemoglobin literature. And so there's also a substantial body of literature that does not acknowledge the existence of this mixed species. And that actually led to a bit of a misunderstanding about this process. You'll recall I mentioned it's the 30% HbF patients that are largely asymptomatic. And so it was assumed in certain parts of the literature that 30% HbF meant 30% of the tetramers contained 2 of the gamma subunits, and then the other 70% were 2 copies of the adult version. But again, a number of studies show very clearly that, in fact, this mixed species not only forms, but the association of the fetal with an adult is equally has an equal affinity as either of the subunits for itself. And so really in a population that is 30% fetal hemoglobin, the distribution of the tetramer species is a simple random probability, from a bag of 30% fetal hemoglobin, pick 2, and that's the distribution that you get. And from that, it turns out that roughly 50% of the tetramers have either 2 or 1 copies of the fetal version. And again, it's also very clear from the literature that this version that contains only the single fetal subunit is also protected from polymerization and does not contribute to polymers. And so again, it's actually 50% in those 30% HbF patients that is protected. And then also to the model, in addition to adding this HbF species, also needed to add the other 2 mechanisms that included binding to 2,3-BPG, so included that using mass action kinetics, as well as binding to a potential oxygenated stabilizer, and here included the possibility of either single or double binding, as both are in development in the clinic. And I'm not going to go through the equations other than to say one can follow the same assumption that if you assume that oxygen binding is always at equilibrium, you can come up with these equations based on a relatively small number of parameters to describe the oxygen saturation of all of the hemoglobin species. And so again, not going into the technical details today, this model, the parameters were fit to oxygen saturation curve data from that was performed under different concentrations of 2,3-BPG. And the model was then validated by showing that it well predicted the shift of the oxygen curve from clinical data of voxelotor, which is an oxygenated hemoglobin stabilizer that's currently approved. And it did a nice job of predicting that shift. I'll also say that thus far, the model has well predicted the efficacy of clinical molecules that are in the clinic for each of these mechanisms. So again, the gene therapy with 30% HbF, as one would expect, this is pretty direct. It recapitulates patients that endogenously express 30% HbF, but also for voxelotor stabilizer, it well predicted the moderate efficacy that's seen with that, as well as there are now a couple different agents that are lowering 2,3-BPG, and the model prediction was well in alignment with what was seen. And so now to get to the answers to the key questions that we were interested in addressing, which was first, by these other 2 mechanisms, an oxy-HB stabilizer or 2,3-BPG lowering, what level of target modulation is required to get to a similar efficacy as 30% HbF? And again, this model is based on the premise that a patient with the same peripheral tissue deoxyhemoglobin concentration is expected to have the same probability of polymerization, which is what's driving disease. And so simply use the model to simulate where that concentration was equivalent with different levels of therapeutic intervention, and found that 51% occupancy by a stabilizer or reducing 2,3-BPG by about 80% is what's expected to give equivalent efficacy to 30% HbF. And then also use the model to consider what level of modulation would be needed in combination. And so that's shown in these plots here, where I should first point out that all combination, or the effect of one can always reach greater efficacy by combining any 2 of these mechanisms, so that if one mechanism is dose limited, combining it with another mechanism will enable reaching a greater level of efficacy. And here in these plots shown here, the peripheral tissue deoxyhemoglobin concentration is shown in the coloring, where white represents equivalent to 30% HbF, red represents greater than HbF or less efficacious, and blue represents less than 30% HbF or expected to be more efficacious, if you will. And so the dotted line here represents every possible combination that could lead to equivalent efficacy as 30% HbF. And then I'm also highlighting with the dashed line some combinations that are likely on the horizon of being achievable based on what's in the clinic right now. So again, voxelotor is already approved. It provides about 25% coverage of hemoglobin. And so that could be combined with 20% HbF or with just over 50% lowering of 2,3-BPG. And this represents a really interesting combination because there are a couple of agents in clinical development that are reaching just about 50% 2,3-BPG reduction. And on its own, voxelotor, as well as these 2,3-BPG reducers, are providing only a very moderate efficacy for patients. But this model is suggesting that combining them together could actually have quite an impressive improvement in efficacy. And I do just want to jump back quickly to a couple of the assumptions that are in the model. When I was developing this model, it was really trying to consider each of these mechanisms of action as somewhat generic. And so some of the assumptions that I made won't necessarily be true for all molecules of the class. And as a couple examples, I assume that a hemoglobin stabilizer binds only the R state. It certainly could be that it prefers the R state, but also has some binding of the T state as well. And so if that assumption is false, not all bound hemoglobin is removed from the polymerization pool. One might think that a T state bound hemoglobin could still contribute to polymerization. And if that's the case, the efficacious occupancy would need to be higher. And just some suggestions that some comparisons of polymerization, ex vivo polymerization as compared to HbF, which has a very well-defined behavior, would help to understand if that's the case for a potential stabilizer. Another assumption that's made in the model is that a stabilizer does not alter the oxygen affinity of the R state. But again, that could definitely be different depending on the molecule. And so if that assumption is false, the bound R state could release more or less oxygen. And one thing that I haven't mentioned is that these mechanisms, by virtue of shifting the hemoglobin equilibrium such that less of it is deoxygenated, by definition, that means that less oxygen is reaching tissues, because if the hemoglobin is holding on to it, it's not releasing it to tissues. And so, all of these compounds all of these mechanisms thus far in the clinic have yet to show any signs of hypoxia, but it's certainly something that clinicians are keeping a really close eye on. These patients are anemic to begin with, so clearly dramatically reducing the amount of oxygen that a red blood cell is able to deliver could, at some point, if you're reducing it by too much, it would be deleterious. And so, in terms of looking at that effect, it's known that there's going to be a therapeutic index that's driven by that effect. But some of a stabilizer molecule might be more or less of a therapeutic index depending on the degree to which it changes the ability of the hemoglobin to release oxygen. So in conclusion, for the sickle cell modeling work, used a really theoretical approach to develop a model of oxygen saturation curves that could be used to compare 3 different mechanisms and really ground them to fetal hemoglobin, for which there is substantial patient data, such that we have a really good understanding of what level of fetal hemoglobin will lead to what level of VOCs and other measures of efficacy. And so that provides a really nice way to be able to then estimate what level of target modulation is needed, either alone or in combination, so that now as preclinical programs are moving to the clinic, they have really clear targets of what they should be aiming for. Additionally, by considering the possibility of combinations and feeling like we have a really good quantitative understanding of that, that enabled us for some of our programs to really lower the bar of what level of target modulation we are aiming for. And so, this has had really big impacts on some of the preclinical decisions of the sickle cell portfolio within Pfizer, both in terms of which programs to prioritize or acquire moving into the clinic, as well as to reprioritize programs based on demonstrating that a program that perhaps appeared that the level of target modulation that was required was out of reach, but could show that actually it was much more achievable in combination, and that represents a valid strategy moving forward. And so with that, I'd just like to acknowledge a few people who helped out on each of these programs, and also, again, providing the references that go into more of the details, and especially the technical details of how these models were developed, including the fitting and validation. And with that, happy to take any questions.
Thank you, Amy. And just a reminder to our audience that you can ask questions at any point as we go along. Just enter them into the questions box. We do have some questions, but I would like to start out with one. How do you define value from the modeling, particularly if it's a negative type of effect where you get a no-go decision rather than a clear go decision or effect?
Yeah, I mean, I think it's always hard because no decision gets made in complete isolation. You know, for that first example that I gave of the Tafamidis follow-on program that led to the decision to terminate the program, you know, as much as modeling played a really large role, of course, there were other factors that always came into play. Yeah, I'm not sure that we ever try to quantify it in terms of a dollar amount. I mean, one certainly could, you know, in that case say, okay, well, the program was at this stage on the day that we decided to terminate it. Had we not had this model and made that decision, we can calculate how far until we would have failed in the clinic and how much that would have cost. I'm not sure how valid that is because, of course, you never know what additional information would have come to light. So in that case, I think we tend to track it more so based off of stage gate decisions. So things like, did modeling enable a termination decision? Did it enable, you know, a program, you know, the ability to declare a candidate molecule or something like that, and kind of more qualitatively track it that way.
Thank you. Okay, from the audience, I'm going to start with a couple of these. They're on the first part of your talk. And the first question is, was it not obvious from the KD values that the second site would be less occupied? Also from the KD values, the drug has negligible binding to albumin in comparison to TTR. Can you comment on this?
All right, let me start with the first question to say, yes, absolutely. The negative cooperativity, yeah, you could eyeball that easily to say, of course, there's going to be more first site binding than second site binding, but we still needed to have quantified numbers to understand, to be able to then do the analysis of the clinical data to say, first of all, do we have enough second site binding that if the second site provides added efficacy, we think we're powered in our study to be able to detect it, but then also to actually quantify what level of efficacy is that second site adding. So yes. And then the second question about albumin, so there is not negative cooperativity, and I apologize if I led to a misunderstanding there of albumin. The albumin, we found that there are four binding sites on albumin, but they're equivalent. And that actually mechanistically makes sense because albumin also binds thyroxine, which is the target, the endogenous target of TTR as well. So we suspect that that's likely what's driving that behavior. But the KD for albumin is essentially an order or 1,000 times greater than the KD for TTR. But the reason that it's still important is that the concentration of albumin is also roughly 1,000 times higher in plasma than the concentration of TTR. So even though they appear to be so far apart that the albumin binding affinity would have, that binding to albumin would be negligible, it actually does play an important role. Yeah.
Okay. In the TTR binding model, the parameter values were fixed. To what extent does this influence the conclusion that binding of one site causes full stabilization?
Yes. So when we when we went to this model to look at the effect of tafamidis, we did fix the KDs. Yeah, I think that's an interesting question, and we, you know, probably should have explored it further to have left the parameters free and fit them as part of this data fitting as well. I think, you know, it just became too cumbersome is the explanation for that. But yeah, definitely should have followed up with some sensitivity analysis at a minimum that, you know, covered the 95% confidence intervals. I will say that the 95% confidence intervals of the KDs themselves were pretty narrow. So I think it's fair to say that there's high confidence that the results would not have changed.
How do you decide whether the model is good enough to inform the decision making?
So for the case of this analysis of Tafamidis, we could ask questions about how well powered our model is. So we could ask questions that said, okay, if the second site provides 20% added benefit in terms of the impact on some of these clinical markers, do we have the power to, would that have shown up in our analysis? So that was one way to gain confidence. In terms of the sickle cell analysis, I would say it's more qualitative. Again, we have a number of examples where the model, if you plug in the values of clinical assets into the model and say, what level of efficacy should we have expected? Oh, it's spot on what the model is telling us. Yeah, I'm not sure that we can do anything that's more quantitative than that. I would also say that second sickle cell model, it's really directed for preclinical development. Whether your target that you're aiming for should be closer to 40% or closer to 50%, that's going to be driven by clinical data and the clinical response that you've seen. But to know that you should be aiming for roughly 50% was still really helpful in terms of, deciding between one molecule or another, or deciding how hard should we try to to develop a molecule that definitely has 50% versus only has 40. We had some understanding of what we expected the additional clinical benefit to be. So again, a bit more qualitative in the analysis, and especially for some of these mechanisms that are either first in class or sort of effectively first in class by aiming for a level of target modulation that's so much higher than what's been achieved before. There's always going to be uncertainty. There's never going to be clinical data for a first in class mechanism. So that's kind of, I think, the best that we can do.
When you stabilize deoxyhemoglobin and prevent the polymerization, is it possible that it will also prevent conversion of the deoxyhemoglobin to hemoglobin and carry oxygen?
Yes, so that's, if I'm understanding the question correctly, the way that we believe this class of molecules works, so they bind to oxygenated hemoglobin and hold them in the oxygenated form such that as the hemoglobin goes through the peripheral tissue, a larger percent of the hemoglobin remains bound to oxygen. Thus, by not being deoxygenated, it's not releasing its oxygen. And so, yes, that is a consideration. I point out that this model was developed based on ex vivo oxygen saturation curves. And so it's really not built in a way that's appropriate to try to use it to better understand oxygen delivery. So I'll just say that that is the known mechanism of this class of molecules. But the model can't help us quantitatively assess that aspect. And again, there's really a lot of clinical data that is showing that at the levels that have been reached in the clinic, these molecules are not inducing hypoxia in patients by a number of different measures. And the other thing to consider is that once you start as you hit therapeutic levels of target coverage, there's an increase in hemoglobin because there's an increase in the total number of red blood cells, that when you prevent polymerization and you prevent sickling, that extends the life of the red blood cells, which leads to an increase in the red blood cells. So yes, any given hemoglobin is less likely to deliver oxygen, but that is somewhat offset by the fact that there is more hemoglobin total. And yeah, just to say that these molecules that are in clinical development, that is something that a very close eye is being directed at that possible impact.
We have a number of questions. We have just enough time for one last question, but we will send all of the additional questions through to Dr. Moody to address on after the webinar. So for our final question, for the hemoglobin model, you mentioned assuming that oxygen is in constant equilibrium with the hemoglobin. Can you discuss the consequences of this assumption?
Yeah, so as I said, this is an assumption that's made in pretty much all of the oxygen saturation models, and it's been a while since I've dug through the literature, but it is well validated by experiments that equilibrium is reached in a time scale of microseconds. So that seems to be a really fair assumption to make. And in terms of how that would potentially affect things, if it is a bit slower, the main way that it would affect things is that you would have to, take more of a mass actions kinetics approach to the oxygen saturation part, which would make it a bear of a ODE model. So I wouldn't be able to use these nice simplifying equations. But I think ultimately, even if it's a little bit slower, that's not going to have a major impact on how the model is working, just because there is an assumption in this model that the oxygen affinity does not change with with drug binding or with 2,3-BPG binding. So the order that those two things happen doesn't really matter. Again, I do think some additional experiments, that really get into this question of to try and separate out which species are really prevalent in vivo would be interesting.
Thank you. We are out of time. For all of the attendees who've sent additional questions in, thank you so much. We will forward them on to Amy um, to converse with you after the webinar. So thank you, Amy. This was a really nice webinar. It's very nice to see how modeling affects decisions, and particularly preclinical decisions. So with that, have a great day, everyone, and we hope to see you all at a future webinar. Again, thank you, Amy.
Thank you so much.